Okay, thanks to everybody for coming. It's my pleasure today to introduce Chuck Fox, speaker today. It's essentially old home week for Chuck. He did his undergrad here in what was then the Department of Zoology. And then after a couple quick trips elsewhere, did his PhD at Berkeley, but really was here for virtually all the time doing that work in Ingalls Lab. Chuck then went on to NSF postdoctoral fellowship at University of South Carolina with Tim Rousseau. He was there a couple of years and was then hired at Fordham University, almost exactly where I grew up, a little bit north of New York City, and then went from there to University of Kentucky, the Department of Ethology, where he is currently. So Chuck is a leading evolutionary ecologist with strong interest in life history, evolution, and insect-plant interactions. We'll be talking about some of that today. And he's also become an impressive sort of mover and shaker in the field. It's really amazing to see. So Chuck is currently the executive editor, is that the right title, for functional ecology, so he has tremendous responsibilities there. He's also edited or co-edited a series of, is it 5 textbooks now on all kinds of subjects, so evolutionary ecology, behavioral ecology, evolutionary genetics, conservation biology, so all sorts of stuff. And he's also on the board of directors for Dryad, right, the data repository institution. So it's great to have Chuck here to talk to us about inbreeding-environment interactions, experimental studies, and the meta-analysis.
All right, thanks, Jay. This is the first time I think anyone's ever recorded one of my talks, so I'm inevitably going to fall off the stage and do something really crazy. This is really exciting to come back here. As Jay said, I was a student here both as an undergrad, and then after I left for a while, I came back and did my PhD work in Hughes Lab, and so there's a lot of people in this room that were immensely influential to me. Art Shapiro's classes, Scott as a colleague when I was in Hughes Lab, of course, Hugh. Michael Torelli, who's the one who told me to go talk to Hugh early on, got me started working with Hugh, and of course Jay. I spent a lot of time measuring insects in Jay's lab because he had a balance that we needed to use. And, anyways, today I'm going to talk about, as the title says, inbreeding see, I'm being recorded, so I'm going to make mistakes like that. Inbreeding-environment interactions. I'm going to mostly talk about some work we've been doing with a model system, this beetle you see here. I'll tell you a little bit about the beetle and why we work on it in just a minute. So I should start off by talking about what we mean when we talk about inbreeding. Inbreeding in the population genetic sense is when organisms are mating with individuals that are more closely related on average than would be a randomly chosen individual in a population. Typically we measure inbreeding by calculating something called an F coefficient or an inbreeding coefficient, which in population genetics terms is now called the probability of being autozygous. There are a number of ways that bring about inbreeding in populations. One of these is that if populations have structure to them, structure meaning that there's a spatial structure where not all individuals are equally likely to encounter other individuals. The individuals that are local to you are likely to be more related to you, so the pool of mates is chosen from individuals that are local and thus more related. This sort of population structure is typically measured in population genetics by statistics such as FST, which you've probably heard used many times. The sort of inbreeding that I'm interested in is inbreeding that occurs within local groups, so non-random mating that occurs within local groups, such as, say, brother-sister matings, which give you an inbreeding coefficient of 0.25, or selfing within plants, which gives you an inbreeding coefficient of 0.5. So this is how population geneticists typically think about inbreeding. In conservation biology, the word inbreeding is used to mean something slightly different, although it has similar consequences biologically. When you have population sizes getting small, organisms are more likely to mate with relatives just because there are fewer non-relatives available in a local area. So you could have organisms that are choosing their mates at random, but still more likely to choose relatives simply because most individuals locally are related to them. It's a different meaning because now we're not talking about choosing individuals that are more related to you than would be a random individual. In this case, it's the random individuals are more likely to be related to you just because the population size is small. So the word inbreeding has come to be used in this literature. It turns out that the genetic consequences of this sort of inbreeding and population genetic inbreeding are basically the same, and so the ecological consequences are more or less the same. So the words are used interchangeably, although the concepts are slightly different. I think I already just said that, so I'm going to skip that. So why do we care about inbreeding? Well, inbreeding commonly leads to something called inbreeding depression. And the depression here just refers to the fact that organisms have depressed fitness when they're a result of an inbred mating than when they're the result of an outbred mating. So this is a figure I lifted from a paper in Nature Reviews a couple years ago that just illustrates this sort of common observation. What you have on the y-axis here is the fitness trait. In this particular example, it's germination success. It could be any sort of fitness trait that people measure. Here you have inbreeding coefficient. So if you look at as inbreeding coefficient increases, a selfing would be a little bit further out on the x-axis here, about 0.5. As the inbreeding coefficient increases, just notice that fitness declines. Germination success, in this case, declines. And that's a really common observation in plants and animals. Inbred organisms do worse than outbred organisms. The reason this is the case is because inbreeding leads to an increase in homozygosity. Homozygosity is the case where you have two copies of the same allele at a single locus, a single gene. Heterozygous individuals are those that have different alleles at a single gene. Well, heterozygosity has two kinds of advantages in organisms. One is heterozygous individuals tend to have alleles that perform slightly differently, possibly under different sorts of ecological conditions, so their optimums are shifted. And heterozygous individuals then could exhibit something called overdominance. They're just better at lots of things because they have more diversity in the alleles, more diversity in their enzymes and proteins and so on. The other thing heterozygosity does, though, is it tends to hide recessive alleles. Recessive alleles are those that are not expressed unless you have two copies of the same allele. Heterozygous individuals, by definition, don't have two copies of the same allele. So if an individual has a recessive allele, but has another allele that's dominant to that recessive allele, then that recessive allele is not going to be expressed. So in populations where heterozygosity is high, recessive alleles aren't expressed very much. It's particularly important when recessive alleles have negative effects on fitness. So take the extreme example of a recessive allele that's lethal. If it's expressed, it kills the individual. Those alleles can be maintained in populations at low frequency because at low frequency they're almost never in homozygous individuals, they're almost never expressed. When you inbreed, you increase the frequency of homozygotes, you increase the frequency of these recessive alleles, and so you increase the costs to the population and to the organisms that are carrying those recessive alleles. They become expressed. This is probably the major mechanism generating most inbreeding depression in organisms, although there's some evidence that overdominance is also important in some circumstances. I'm not going to talk about outcrossing depression, but I just want to acknowledge that in some organisms, actually there are benefits to mating with individuals that are related to you, especially when organisms are locally adapted. So if you're adapted to soil types that are very, very local, mating with an individual that's unrelated to you might bring in alleles that don't actually perform well. And so selection could favor mating with individuals that are related to you rather than unrelated. There's a little bit of work in plants looking at this sort of thing, but it's not something I'm going to talk about today. Okay, so the main question I want to ask today is, does inbreeding depression vary with environmental conditions experienced by organisms? And well, we know that gene expression varies with environmental conditions, and inbreeding is just a consequence of differences in gene expression between homozygotes and heterozygotes, or difference between inbreds, which are more homozygous, and outbreds, which are more heterozygous. So it makes sense that inbreeding depression should vary with environmental conditions, and in fact there's now a growing number of experimental studies. I'm doing another meta-analysis now, and we've got like 75 or 80 studies in our data set so far. So fairly large number of studies that are manipulating environmental conditions, showing that inbreeding depression varies. But there are a lot of things we don't really understand yet. So we don't understand sort of what sorts of environmental variables affect inbreeding depression, whether novel environments, the environments that organisms haven't experienced in their selective history, have more effect than do generalized stressors, the kinds of stressors organisms regularly experience. What are the mechanisms underlying these interactions between inbreeding and the environment? These are generally not understood at the moment. Why should you care about this? Well, I don't like to put my work in a global change context, even though a lot of the experiments I'm going to talk about are manipulating temperature. And the reason is, the experiments I'm going to show you are going to manipulate temperatures quite severely. We're going to give animals very, very cold environments, very, very hot environments, not the sorts of variation that is directly relevant to global change, climate change, I think, but is really relevant, I think, for getting at what sort of generalized stress, the kinds of stresses that organisms experience, and the kinds of stresses that are particularly experienced in conservation context. When organisms' population sizes are declining, they're now in less optimal habitats, often edge habitats, habitats that are stressful for a variety of ways. And so inbreeding depression is a big issue in conservation biology, but those organisms are typically stressed in other ways other than just being inbred. So, for example, there's a lot of people who are interested in measuring inbreeding depression in organisms of conservation significance and then extrapolating from experimental studies or zoo studies to what's going on in nature or what's going on in other populations. And the only way we can do that is if we have some idea of how environmental conditions affect the expression of inbreeding depression or genetic load. Inbreeding affects the risk of extinction, but those effects are going to depend a lot on environmental conditions. If inbreeding in particular depends on, the consequences of inbreeding depend on environmental conditions. There's a little bit of a theoretical data that show that inbreeding not only accentuates the risk of extinction, but can cause feedbacks with declines in population size that could vary with environmental conditions. Of course, I already mentioned this. Stress is particularly common in organisms of conservation significance. In particular, inbreeding stress interactions can exacerbate the effects of small population size. And then there's a fairly large discussion in the conservation biology literature about purging of the genetic load. The general consensus is that the experiments or the breeding designs that are necessary to purge the genetic load in a population, say in a zoo or in a laboratory environment, before releasing those organisms back into nature, those experiments are probably just not practical. The consequences for the populations, there are a lot of negative effects. But if we're going to be pursuing strategies for purging the genetic load in species of conservation significance, we need to understand whether purging the genetic load in one environmental context has any effect on the genetic load in other environmental contexts. And as you'll see from some of my data, that the environmental effects can be quite substantial. Okay, so here's what I'm going to do today. I've given you a brief introduction to the topic I want to talk about, inbreeding-environment interactions. I'm going to run through a series of slides fairly quickly, just demonstrating what my model system is and that there's a lot of inbreeding depression in this particular model system. I'll work through the first slide with you and then sort of go through a bunch of slides pretty quickly just to make a point. And then I'm going to talk about how environmental variation, in particular temperature and diet, influence the expression of inbreeding depression in my study system. And lastly, I'm going to talk a little bit about senescence in beetles and lifespan and how inbreeding depression and inbreeding load for lifespan is influenced by primarily temperature, but also I'll talk about a couple other variables at the end and hopefully won't run out of time. Okay, so this is the beetle I work on. Here's an actual photograph. This is a female Callicebrucus maculatus. It's a seed-feeding beetle. I actually started working on it when I was in graduate school here, and Hugh's lab was inundated with loose beetles flying around for a couple of years. Very tolerant of my lost beetles. It's a really great system to work on because the beetles are seed beetles. Their whole life cycle revolves around seeds. This is a Nazuki bean, one of many species we work on. These little white dots here are eggs that are glued to the outside of a seed. Females glue the eggs to the outside of the seed. The egg hatches, larvae burrow into the seed, eat the inside, go through all of development, pupate, and then emerge as an adult from an exit hole like this. Adults don't need to be given access to any food or water or anything except seeds. So you never have to feed them, never have to change out vials of sugar water and the moldy vials that you get, the moldy cotton in the lab and so on. No, you don't have to do any of that. Just throw beetles in with seeds, they lay eggs, they develop, you can do stuff with the adults. So it's a really easy system to work on. This particular species is an agricultural pest. We work on some species that are not ag pests. We've done some overlapping work, but most of our really good data is on the ag pests, so that's what I'm going to talk about. They use a lot of species of beans. They don't use everything in this picture here, but this is a poster that I actually got also when I was here at Davis in graduate school, illustrating the diversity of many kinds of beans that are grown in the United States. If you zoom in here, open it up, you get a picture or lots of the different kinds of beans. I'm going to talk about black-eyed beans a bit, cowpea, which is the sort of the broader group. Black-eyed beans are a particular variety. But if you zoom in even further, you notice that the beetles attack a number of different species here. They've attacked these, they've moved to these, they get the cowpea up in the corner. So they use a lot of different hosts. So you're supposed to laugh at that slide. Didn't see that, at least laugh now. So. All of my experiments are going to use a very specific type of experimental design. This is something I call a block design. The block design is basically where we're going to cross two families. So let's take family A and family B. They're both unrelated, outbred families. We're going to take a male from family B and cross with a female from family A and vice versa. These are going to be outbred. And then we're going to do a cross within each of the families. So a female with a male from family B and the same from family A. This is not the sort of design that's typically used in the plant literature, the plant ecology literature. The typical design in the plant ecology literature is to take a single family, say family A, self the individuals to create inbred, and then mate to some male selected that's unrelated. The problem with that design is that the inbreds and outbreds don't just differ in heterozygosity, they also differ in the source of pollen. So there's lots of different alleles that have been brought in to the design and are frequently not controlled for because often they just use a handful of males from the population. What we're doing here is because we're doing all our crosses just within two families, there's only a handful of possible alleles present. And they, on average, not for every individual beetle or family, but on average, the alleles that are present in outbreds are the same as the alleles that are present in inbred. They just differ in whether they're more homozygous or less homozygous. So that's our basic design. Every single block gives us a single data point, which gives us means fitness for outbreds and inbreds, and it gives us a measure of inbreeding depression. So all of our experiments are made up of lots of different blocks, each block being a single data point, typically 40 or 50 blocks per treatment or experiment or whatever. Now, throughout my slides, I'm going to describe inbreeding depression in two different ways. It's sort of unfortunate, I know, that each of my slides is going to switch between different metrics of inbreeding depression, but the reason is, if you're trying to publish papers on inbreeding in ecological journals, people like to see you describe inbreeding depression using this variable delta, which is just the proportional reduction in fitness. So an inbreeding depression of, say, 0.2 means that inbreds have 20% lower fitness than do outbreds. 0.3 would be 30% lower fitness. If we were talking about development time, 0.2 would be 20% longer development. Something like that. Fairly straightforward. Just the proportional difference in fitness between inbreds and outbreds. In genetics and evolution journals, more often people calculate something called the inbreeding load. The inbreeding load is actually a number that is sort of what would need to be true in our population if we were talking about lethal alleles segregating in a haploid population of organisms, which of course is not what our organisms are. They're not haploid, we're not talking about lethal alleles, but it's the equivalent number of alleles that would need to be segregating in an average individual if they were lethal and the organisms were haploid. So the numbers themselves are not estimates of real numbers of genes involved. They're estimates of what would be an equivalent number of genes that would generate the same data if these conditions were true. A lot of the data I'll show you has lethal equivalents ranging from anywhere around half or so up to, in some cases, 8 or 9. So that would be a very, very high genetic load or a very high amount of inbreeding depression. The two variables are completely correlated with each other. So, and they're not completely linear, but they're not that far off from linearly correlated. So when I switch between these, don't look at the absolute values. The absolute values here will be easily interpretable, not quite so here, but the relative magnitude across treatments will be basically exactly the same regardless of what metric I show in the figures. Okay, so back to my study beetle. How much inbreeding depression do the beetles exhibit? I'm going to show you a lot of slides that look sort of like this. What I've got in this particular slide is a fitness trait. In this case, this is proportion of offspring that survive through different periods of development across the different stages of development. So the first stage here is whether eggs develop or not. Solid circles are outbreds, open circles are inbreds. And you see that inbred eggs laid from sib matings are a little bit less likely to hatch, maybe 4 or 5% less likely to hatch. Top panel is one population, bottom panel is a different population of beetles. All of my experiments are done on these same two populations of beetles, and so there's always two sets of data on every slide I'll show you. So eggs are a little bit less likely to develop at all, meaning the embryos don't even form if they're inbred. If they develop, they're still less likely to hatch. So some of those larvae are dying as pre-hatch. If they hatch, they're about 10 to 15 or so % less likely to reach adult. And this last column here just compares across all stages of development. You get about a 17 to 20 or so % reduction. If an egg is inbred, it is about 17 to 20% less likely to produce an adult beetle a month later than if it's outbred. So, fair bit of inbreeding depression in survival. If the beetles survive to adult, inbreds take longer to reach adult, about 5% than do outbreds, so this is development time. So inbreds take longer to reach adult than outbreds in both sexes, both males and females, again, two populations. If females reach adult, females have about 20% reduction in fecundity if they're inbred relative to outbred. If females are outbred, but they mate with an inbred male, they have lower fecundity than if they mate with an outbred male. This is actually two separate experiments here. We had to repeat it because we didn't believe it. You got two separate experiments. All of these are outbred females. These are females mated to outbred males. These are outbred females mated to inbred males. Significant reduction in fecundity in every single case. The reason for that is probably that inbred males produce less sperm than outbred males do. They don't produce smaller nuptial gifts, but they do produce less sperm. Okay, so that's sort of a basic overview of the system. You'll notice that there's not a lot of inbreeding depression on any individual trait. Often they're 3%, 5%, sometimes as much as 15 or 20%. But if you add it up across all the different things that contribute to successful survival and reproduction, inbred beetles are pretty screwed. They don't do very well at all. If you add everything up, they just don't survive well. They don't make very many kids compared to outbred beetles. I started working on how temperature and diet affect inbreeding depression when I had a graduate student who was working on adaptation to temperature, one student, and I had another student who was working on inbreeding depression in the beetles. And so we decided to put the two experiments together. Let's just manipulate inbreeding, let's manipulate temperature and see what happens. And this is just one example of what some of our data look like. What I've got here is I'm just going to show you one fitness variable. This is just the probability that an egg that's laid produces an adult. What you've got here are two populations of beetles reared at 4 different temperatures. Again, the reason I don't talk about this in a global climate change context is we're talking about a really large range of temperature. You get much below 20 degrees and beetles are really stressed. You get much above 35 degrees and beetles become really stressed. So this is sort of a reasonable range where outbred beetles do pretty well. Above and below, they don't do nearly as well. Solid circles are survival of outbred beetles, open circles, survival of inbred beetles. And there are a couple points that I'll make. The main one from this slide is that You'll notice that as we get higher temperature, outbred beetles have reduced survival, but they actually don't seem to mind some lower temperatures. And this comes up in a lot of experiments. You reduce temperature and outbred beetles don't mind it all that much, but inbred beetles mind it a lot. They don't do well at low temperature. I'm going to show you a bunch of things in our data set that show that low temperature really changes up things compared to normal temperatures. So my first point, and we have a few data sets that support this, is that whether an environment is stressful or not for development of our beetles depends on whether they're inbred or outbred. You can raise outbred beetles in a much larger range of environmental conditions. When you get to the extremes, inbred beetles start feeling it much before outbred beetles do. Second point I would make is that from these data you can calculate estimates of inbreeding depression or the inbreeding load, and you'll notice that they're highly variable across temperatures. At intermediate temperatures, inbreeding depression is lower. When you get especially to cold temperature, inbreeding depression gets really, really high. So lots of variation in inbreeding depression across temperature. Here's another experiment where we manipulated temperature and diet at the same time. You see the same thing. Intermediate temperatures, the beetles do well. Inbred beetles don't do well at low temperatures in particular, and you get a lot of inbreeding depression and a slight increase of inbreeding depression at high temperatures. But temperature has a large effect on the expression of inbreeding depression. And notice that here we got inbreeding depressions of like 70 or 80%. That means they have about 20 to 30% as high of survival, just one fitness trait, compared to the beetles that the inbreds compared to the outbreds at that temperature. So a huge amount of inbreeding depression.
I'm having trouble distinguishing inbred versus outbred and cowpea versus mung.
Okay, so this is a plot of just inbreeding depression. So there's no inbreds and outbreds on here. This is the proportional reduction in fitness of inbreds compared to outbreds. Sorry, the slide axis changed between those figures. Good question. The open circles here are different diets. This is cowpea. This is mung, two of the common diets we use. Beetles don't do as well on cowpea as they do on mung. They still do pretty well on it. And there's a little bit of evidence here of a diet by temperature interaction, but it's mostly driven by one population. It's actually significant in both cases, but not a real huge effect. Well, one of the questions that I think is really interesting is what's going on genetically that's generating these differences in inbreeding depression between different environments. And clearly that's the sort of question that's in the realm of genomics, and there are people that are starting to ask these kinds of questions using genomic techniques. We tried to ask these questions in our data set by taking advantage of the fact that we had these experimental blocks I described earlier, where each block has inbred and outbred beetles. We can calculate an estimate of inbreeding depression for each block. We also, what I didn't say is that in many of those experiments, they're split family experiments where siblings are raised in all temperatures. So we can look at whether sibs raised in one environment exhibit the same amount of inbreeding as those same family members would in another environment or another environment, and so on. So what these are plots of Pearson moment correlations, so just standard correlations between inbreeding depression in pairs of environments, where in each case we're comparing sibs in one environment with sibs in another environment. Those are our data points. And I think there's two things you see from this. One is that all of the estimates are positive. That tells us that at least to some extent the genetics underlying inbreeding depression in one environment are probably similar to those same genes, similar effect sizes in other environments. But you'll also notice that a lot of them are not very high. The correlations are often pretty low. Some of them are quite low, like 0.2 or so. And I think that tells us that although there's similar genes expressed in each of the environments, there's also possibly a lot of different genes that are contributing to inbreeding depression in different environments. Or if they're the same genes, the magnitude of effects are changing quite a lot with environmental conditions. So that's what those 2 points say, what I just said. Okay, so one of the big questions in the literature is, to what extent is inbreeding depression and its variance among environments simply because animals or plants are becoming more stressed? Is it a consequence of being stressed? Unfortunately, most of the studies that are published are used maybe 2 different environments, one of which is benign, one of which is more stressful. And it's really hard to determine sort of any general relationships when you only have 2 environments to compare. So using our data, we have multiple environments. We have 4 temperatures, 2 different diets, so at least 8 combinations of environments. We have 2 different populations of beetles. So we wanted to ask, in a situation like that, can we detect a sort of general pattern between stressfulness of the environment and the magnitude of expression of inbreeding depression, or the genetic load? So what this figure shows you is a plot of what we call just amount of stress, where stress is a measure of the degree to which outbred beetles do worse in alternative environments compared to the best possible environment. So let's say that our beetles have 100% survivorship at 25 degrees, the really good temperature on mung bean. They don't have quite that high. They have really high survivorship. So let's just say it's 100% survivorship. That would be our most benign possible environment. The beetles do great on it. And a stress of 0.2 would be an environment where outbred beetles survive 20% less. They do 20% worse. A stress of 80% or 0.8 would mean that in this environment, outbred beetles have an 80% reduction in survival compared to the best possible environment. So that's our index of stress. And you'll see that it actually correlates fairly well with the genetic load expressed in those environments. The genetic load expressed, or the amount of inbreeding depression in good environments, is fairly low. As environments get more stressful, you get a substantial amount of genetic load being expressed. So this axis here is the genetic load for this period of survival between an egg being laid and whether that egg produces an adult offspring. Positive relation. Actually, a very steep slope. Notice that the genetic loads are above 8 or so, which is a phenomenally high genetic load in the more stressful environments. Well, when I did this project, I was collaborating with a guy at University of Louisville, just down the road from me, David Reed. And David Reed had written a review paper on basically how does the environment affect inbreeding depression. And so it's a catalog of studies. Basically, environment matters, but they couldn't really find any general patterns. So we took his data set that he compiled, and we did this exact same analysis. So this is an analysis from a few years ago of all of the published studies that have at least 2 environmental conditions. The benign one, by definition, is the one of those combinations of environments that the animals or plants do the best at. The fitness variables vary a lot. Some of them are measuring numbers of flowers produced, some are measuring survival, so there's a lot of heterogeneity in the fitness traits that are being measured. So good environment, the one the organisms do best at. Stressful environment, the one they do less well at. We measured stress in the same way I described in the previous slide. This is the relative fitness of outbreds compared in the poor environment with the good environment. The relative reduction in fitness, I should say, in the poor environment compared to the good environment. And then we looked at the genetic loads in those environments and how much they changed. So this is the change in genetic load between those pair of environments, and you find this nice positive relationship. The more stressful the stressful environment is compared to the benign environment, the greater the difference in genetic load between those 2 environments. Nice positive relationship. It's a slope here that is for about every 30% decline in outbred fitness, 30% stress, you get an increase of about 1 lethal equivalent. So a relatively steep slope. Not as steep as in my experimental study I showed you a second ago, but nonetheless fairly steep. After we published that paper, that was published in 2012, while that paper, I guess, was going through review, Laramie Enders and Len Nunney from UC Riverside did the same analysis on some Drosophila data. And in their Drosophila data, this is an overlapping set but not identical set of studies. They found some that we didn't know about, and we had a lot more than Drosophila. They got exactly the same relationship in their analysis. The slope here is a slight bit lower than the one in our analysis, but not a lot lower. It's a little bit lower. So to summarize the first part here of the talk, inbreeding depression in my animals, the beetles I work on, clearly varies with temperature and diet. There's a little bit of evidence that stressors interact with each other, although in our particular case, temperature and diet don't interact a lot, but they interact a little bit. I think the three sort of main conceptual points is that whether an environment is stressful or not for beetle development depends on whether they're inbred or outbred. In particular, low temperature is not very stressful for outbred beetles. They can tolerate low temperatures, not exceptionally low temperatures, but much lower temperatures than inbreds can. As you reduce temperature, inbred beetle survivorship drops off very, very quickly. Inbreeding depression is affected at least by at least some overlapping loci among environments. So those correlations I showed you between blocks, the between block correlations compared across temperatures. We've also done it across diets. We've done it in a number of experiments, and the results are always basically the same. All the correlations are positive, but they don't tend to be very high, which suggests that there's a lot of changes in gene expression that are occurring across environments, but there's also a lot of commonality in the genes and the expression of those genes in pairs of environments. And then inbreeding depression clearly increases with environmental stress. We saw this both in my beetle study, but we saw it in our meta-analysis as well, and our meta-analysis includes lots of different kinds of organisms, different types of fitness measures, and so on. In general, we see an increase of about 1 lethal equivalent for every 30% reduction in outbred fitness, or every 30% reduction or increase in stress. Okay, so why does inbreeding depression increase with stress? There's a lot of different kinds of hypotheses proposed in the literature. They're not alternative hypotheses. They often come from very different perspectives. So for example, there's a population genetic hypothesis that exposure to stressful environments alters the genetic architecture underlying inbreeding depression. So for example, you can have a change in selection coefficient across environments from being, say, weak selection against recessive alleles in benign environments to being strongly negative in stressful environments, or you could even have alleles that are under positive selection in benign environments be under negative selection in more stressful environments. There's a little bit of experimental evidence that support this, but I don't know that there's a good population genetic argument to support this, why you should have directionality in the change in selection. And Michael Torelli is here, so maybe he'll tell me I'm wrong on that, and I hope not. There's a physiological set of explanations, basically, that the expression of genetic load increases the sensitivity of inbreds to the physiological effects of environmental stress. So for example, you can have breakdown of the heat shock response or breakdown of other generalized stress responses in inbreds compared to outbreds. There's actually a lot of experimental data on this topic and generally supportive, although we're still sort of in the infancy of understanding that. The newest set of ideas has to do with epigenetic control. Of course, epigenetic control of gene expression is a huge topic nowadays and is involved in how organisms respond to environmental conditions. And so there's been a proposal that inbreds basically have screwed up epigenetic control of gene expression, and so they can't respond well to environmental conditions. They do poorly under stress. And then what I just want to talk about a moment more in the next slide, because it's a statistical argument, is that the increase in inbreeding depression in stressful environments is a statistical consequence of the increase in phenotypic variance caused by stress. So Don Waller proposed in 2008 that if stressful environments cause an explosion of variance within a population, and they increase phenotypic variance. Phenotypic variance sets a limit to how much natural selection can occur in a population, and inbreeding depression is effectively a measure of selection. It's the degree to which one type of genotype does worse than another genotype. So this change in phenotypic variation should affect inbreeding depression in the same way that it can potentially affect selection on other traits. Fortunately, this is the kind of data we had, and we could ask this question. I got together with a number of colleagues, David Reed, Torsten Christensen, and some others. We got all of our data sets together, and we asked, first off, does the coefficient of variation in fitness increase with stress? And the answer is generally yes, it does. So this is across all studies. This is for my particular study. Really high correlation between variance in the fitness traits we're measuring and how stressful the environments are. Is that adequate to explain why inbreeding depression increases with stress? In 4 of 9 of the data sets, stress, independent of the coefficient of variation, is the best predictor of the genetic load expressed in populations. In 4 of the 9, both stress and the phenotypic variance are in the best fit model. I think this is where mine comes out. And in 1 data set, the coefficient of variation in fitness is the best predictor of inbreeding depression in a population. So it seems that yes, this hypothesis, Waller's hypothesis, is a good one, that it may be a function of the increase in variance in fitness that predicts some of this relationship, but that doesn't tell us mechanistically what's going on. Still ecologically important, there's still underlying physiological and genetic mechanisms to understand, but statistically this relationship does exist. And this is just a combined model that shows you that stress in all of the data sets combined is still the best predictor of inbreeding depression, but that the phenotypic variance increasing with stress is a major contributor to understanding what's going on in these studies. Okay, I'm going to switch gears a little bit and start talking about, I know I don't have a lot of time left, so I probably won't get through all this, but I want to talk about one of the other themes I've been working on and how it intersects with what I've talked about so far. I'm really interested in senescence and why senescence varies among populations, why it differs between males and females, and that kind of stuff. So we've done a lot of work looking at lifespan and patterns of senescence and mortality rates and so on in our beetle populations. I'm going to boil this down to just talking about one of those traits, which is lifespan, although we see it in all of the other ways we measure mortality in our populations. So we discovered quite a few years ago in one of my graduate students' projects that if you measure inbreeding depression in adult lifespan, again, we have 2 populations of beetles. Here's outbreds and inbreds females and outbred and inbred males. You notice that there's a significant reduction in survival adult lifespan of inbred females, but no difference between inbreds and outbreds in males. Same thing in the other population. A fairly large difference in the magnitude of inbreeding depression from more than 10% to effectively 0 between females and males. So something interesting is going on there. It turns out if you compare populations of beetles that are long versus short-lived, what you observe is a lot of dominance in hybrids between when you cross long and short-lived populations of beetles. Notice that all of these points are above a line. This is the line of additivity. This is due to there being a lot of dominance of long-lived alleles over short-lived alleles. Well, inbreeding depression is a function of dominance. It's that interaction between the 2 alleles at a single locus. If you're heterozygous, one allele is not expressed as much as the other allele is. This is what you get in females, lots of dominance. In males, you don't get any dominance. The dominance effect is nearly 0. The reason some of these points are above the line and some of these points are below the line is there's a large maternal effect. If your mom is long-lived, the kids tend to be long-lived. If mom is short-lived, the kids tend to be short-lived, but there's no directional dominance there. So consistent with the difference in inbreeding depression between males and females. I'm going to skip this slide. This is just sort of a set of possible explanations to explain the sex difference, but I don't really have time to go through them. So we've been, of course, manipulating environmental conditions and asking how the genetic load changes, and so we said, well, does it matter for adult lifespan? So here's sort of the kind of data, this is a completely different experiment, but the kind of data I just showed you. This is the genetic load for females and for males when they're reared at sort of standard laboratory conditions, the conditions they do really well at, they really like. 2 populations. What happens when we manipulate temperature? Well, as temperature declines, you get an explosion of genetic load in males and the sex difference goes away. Notice it happens in both populations. As you increase temperature, we get sort of an inconsistent change. Generally, inbreeding depression in males goes up, but it's not consistent in every experiment that we do. But what is consistent in every experiment we do is that inbreeding depression in males increases dramatically at low temperature and the sex difference goes away. So this big effect we've seen in our standard lab experiments changes dramatically as we manipulate rearing temperature. This is temperature of animals that are from the moment the egg is laid, say within 12 hours of egg laying, until the beetle dies, they're at one of these temperatures. They never get switched. If we just manipulate adult temperature, so they're all reared at a common temperature, when we manipulate adult temperature, basically nothing happens. The inbreeding depression is much lower in males than females. Not much change with temperature, maybe a little bit of dip here in females, but not a huge change with temperature. In contrast, if you manipulate rearing temperature, so that the beetles are reared at different temperatures, but then moved as adult to a common temperature, so everybody lives out their life in a common 26 degrees where they're very happy, this huge increase in genetic load in males. Something about being reared at low temperature causes an explosion of expression of the genetic load in males, and the sex difference completely disappears in both populations of beetles. That got us wondering about other sorts of things that we do in the lab and how they might matter. So for example, all of my beetles, when we do these experiments, we use virgin beetles. As soon as you make females, you have all these complexities about how many eggs did they lay and what are they doing, and all that affects patterns of senescence. So we just look at virgin beetles. It makes everything really simple. If you mate females, their genetic load, so these open circles, two populations of beetles, mated, the genetic load for females that are mated versus virgin, genetic load drops dramatically and goes to about 0. When females are mated, their genetic load is not any different than it is for males, and it's very different than the genetic load expressed in virgin females. We also don't feed our beetles in the lab. We don't think they eat much in nature. There have been a number of studies in this. Beetles will go to flowers and drink nectar, but most of the time they don't have that available. If you give them access to various kinds of diets, you get a very small increase in fecundity, a few percent, 5, maybe 10%. You do extend their lifespan though. If you give beetles access to sugar water as adults, here's males that have no access to sugar water, our standard laboratory conditions. See the big sex difference here. Give them access to sugar water, and all of a sudden, genetic load is expressed just like it is in females, in two different populations. Big effect of diet on males, but not on females. Mating affected females, but not males. Diet affects males, not females. So what the heck's going on? Well, so I'll wrap up here. I'm out of time. So we know there's a big sex difference in the genetic load affecting adult lifespan of our beetles. This sex difference in the genetic load for lifespan depends on temperature. There's a large rearing temperature effect, but we don't seem to see any adult temperature effect. What they're living their lives out at as adults doesn't matter, but the rearing environment they experience has a large effect. So what kinds of genes are turning on at low temperature in males, but not in females? Or what genes are turned on for females across all temperatures, but only at low temperatures for males? Those are the kinds of questions we need to address. And then lastly, the sex difference in genetic load for lifespan depends on both mating status and on diet. So there's got to be something going on with sex differences in resource allocation that's influencing expression of the genetic load. There's a possible role for insulin signaling. Insulin signaling is a big deal in the senescence literature, and so it's possible that our diet is triggering the insulin signaling response, and that is different in inbreds and outbreds. So I guess I will stop. I know there's a class coming in a minute, so let me just jump to my acknowledgements. There's a bunch of people here that worked with me on this. Christy Shively was a PhD student in my lab who started a lot of the inbreeding work. Craig Stillwell was working on adaptation to temperature, and it's sort of the merger of their projects that led to what I'm talking about today. Bill Wallen was my technician for many years. Jingjing here taught us how to measure sperm, and I always like to tell the story that she worked on tribolium, and she counted sperm and tribolium. when she came into my lab to work with my beetles, which are about 5 milligrams, so pretty small beetles, she looked at them, and the first thing she says is, wow, they're so big. I don't get it, but. And then David Reed, who a number of my papers are co-authored with. David is faculty at University of Louisville. Sadly, he died about 2 years ago, and he was a great collaborator and contributor. I'll stop there. Thanks.
So we have just a minute or two for questions for Chuck.
Genetic architecture, come on, that was a joke. So why do deleterious alleles just become more deleterious in the changed environment? You have more homozygosity, and you just estimate a dominance coefficient. Nothing about changes to
Yeah, so I mean, there has to be some mechanism by which it could be just the dominance, the degree of dominance is changing, but there's got to be some intermediate, what's happening with gene expression to generate the phenotypic differences.
Why the amount of dominance change? They have more homozygous.
Oh, no, our homozygosity isn't changing with stress. We're controlling homozygosity, so it's the same in every treatment. So, yeah, there's no difference in homozygosity. The F coefficients are the same. Yeah. So when we're increasing, say, stress, we're changing the temperature that the animals are reared at, but the treatments are identical in each case. So homozygosity is the same.
The deleterious effects.
Yes, the deleterious effects of increase in homozygosity are increasing. Yes.
How much of the effect of low temperature is the actual effect of temperature on slowing development? Are they just not making key developmental stages fast enough or something? And they're integrated messed up?
Yeah, so one of the hypotheses that's possible is that we're getting non-random mortality, and so non-random mortality in general. So maybe males are just dying at a greater rate than our females, and that's generating different genotypes in the adult population that generates the sex differences we see, because we effectively weeded out particular groups of males but not females. We don't think that's the case because the sex ratio doesn't change. So it is possible that you're still getting non-random mortality, but it's not like lots of males are dying and not females. At least it's not that. The genetic sex ratio is very close to 0.5, and the emergent sex ratio is really close to 0.5. It varies a little bit across treatments, but not enough to explain the differences we see.
Let's take one more from Scott, and then we'll...
Scott.
Zooming out just a little bit, does inbreeding biology have particular relevance to calciferous in nature?
Well, I say so in my grant proposal, so I hope so. So the last grant proposal I got funded, we focus on, and I think this is real biology of the beetles. These are the stored products pests. So the population sizes are giant in nature, and in agriculture fields are going to be large. But when crops are harvested, they're colonized then by females, and so the population sizes immediately following colonization are necessarily small. And so the biology that occurs during that period of time should be very significant for the population dynamics of the animals, yes. So it's relevant in this case because of the stored products context. It is true, though, that a lot of these brucids in nature, this one and the other one I work on, are gigantic population sizes in the field. And so you wouldn't expect in wild populations them to be under, to have experienced a lot of inbreeding and a lot of inbreeding depression. But colonizing of novel habitats, colonizing of seed resources. We've got one population in Arizona where there's a few isolated trees, and there'll be no fruit or beetles there for a few years. You come back later and everything's fruiting and they've been colonized. So there's clearly a period where the population sizes are necessarily small.
Thanks very much, Chuck.
All right, thank you.